Yvonne Lawley is an associate professor at the plant science department at the University of Manitoba. Her area of research is agronomy and cropping systems and has focused on several crops, including soybeans, corn, and wheat, and a range of management practices, from residue management, strip tillage, to cover crops. Her research includes small plot and on-farm field-scale agronomy research, and she shares the results to a wide range of audiences, including farmers, agronomists, scientists, and in the classroom, where she importantly teaches students about the fundamentals of cropping systems. So Yvonne is going to talk today about crop rotation under the microscope and discuss a lot of the work that our commodity group and other commodity groups have funded her to conduct research in over the last number of years. With that, Yvonne. This is the pointer and then the box.
Thanks, Daryl. Well, good morning, everyone, and thank you for the invitation, Daryl, to speak this morning about some research that I think is really interesting that relates to soil health. And one of the goals I have this morning is to bring a collection of things together to talk with you about what we've been able to learn in my lab at the University of Manitoba that connects important management practices that revolve around crop rotation that can contribute to soil health. One of the things that we're learning at the same time as we're looking at these management practices is about soil health itself and how we can measure, quantify, and understand soil health in our prairie soils that have so much to offer. So I hope I'm going to, as the first speaker, lay a bit of foundation that we're going to be building this whole session off this morning. So when I was preparing for this presentation, one thing I was reflecting on, if any of you have been at the University of Manitoba, maybe we're lucky enough to have Gary Martins as a professor, and I took crop production from Gary, and I remember when I was studying for his exam that I just had to remember crop rotation. If I could write about crop rotation, I was probably going to at least get partial marks for whatever answer I had on Gary's exam on my crop production final. So I think that this presentation today that revolves around crop rotation and crop rotation studies, again, is sort of a central element. If you're putting together that award-winning answer for your farm on building a soil health strategy, crop rotation is going to be at the center of that answer. So because I'm first and because I'm a professor, I wanted to talk a little bit about fundamentals. Let's just make sure we all are clear on what we're talking about, soil health. And I have this wonderful picture from my graduate student, Jacob, who took this picture in one of the long-term experiments at the University of Manitoba. To me, this looks like a healthy soil. We've got healthy plants living, we've got some residue protecting the soil. The goal of soil health is to continue to do the things that we rely on soils to do, for it to function as a living ecosystem that sustains all of the things that we care about, humans, animals, and the plants that make us all function, capture the solar energy that we need to be able to grow things that are important to us as humans. We rely on soils when they're functioning well to do a lot of things, including, in an agricultural setting, we're almost always focused on how soils support crop growth. But as Daryl mentioned, we also rely on soils to do a lot of other, have a lot of roles, and as society in general is turning to farms and to farmers to support these functions in how you manage this important resource that we have, which is the soil. It also connects, as Matthew talked about in that video, with water and how we rely on water in this province and water quality. So what do we have to go on if our goal is to improve soil health? Many of you who've been reading about soil health have probably seen this list of 5 soil health principles in many different places. And today I want to touch on these 5 important principles through the lens of different studies that we have at the University of Manitoba that connect to the concept of using diversity, crop rotation, living roots, soil cover, limiting disturbance, and where we can, integrating livestock. What tools do we have to know how our soil is functioning? This is where there's a lot of research going on right now, and especially bringing tools that other places other regions have been using and testing them out here in our soils in Manitoba to understand which tools are going to be useful to us to identify soil management practices that are actually making a difference. Soil health is a complex landscape, and it's really important to think about which soil health goals you're wanting to achieve on your farm, to connect those with these different monitoring strategies. So these different dimensions that we can talk about soil health relate to physical, chemical, and the biological properties of soil. And Steve's going to lay out, I hope, in our panel presentation, a few promising methods that he's been utilizing and calibrating with soils in Manitoba. Matt is going to talk with you, Matthew Bacher is going to talk with you about some of the biological elements of soil health. That's an area he's working on in his lab. We're also going to learn a little bit about the fine balance between the good actors and the bad actors as we talk about creating an environment that's both enabling good soil health, good biology in the soil, as well as maybe suppressing or helping us to manage pathogens in the soil with Sima Chatterton. Charlie's going to talk to us a bit about the physical dimensions of soil health, and I wanted to just lay out a few things related to soil and soil compaction. That's a topic I'm really interested in. We have this beautiful picture here of a healthy soil that's supporting one of the microflora that we can actually see, earthworms that we think about, and plants and soils require space in soil. And so we need to think about creating that home for them, and we often think about soil as this mineral component, right? If we think about what soil is made up by volume, yeah, there's this mineral part that provides nutrients, that has all these chemical properties that we rely on. But half of soil is also space. Space for air, for these microbes to breathe, and for roots to get the air that they need. And there's also room for water, and how we want and need water to be a part of this dynamic component. So when we're talking about soil compaction with Charlie, we're going to be thinking about how we preserve the space inside of soil. We can preserve this space by managing how we traffic soils. And I wanted to show you this picture here. Again, any of you who have been at the University of Manitoba campus and maybe have relied on public transportation have made this dash across the quad to catch your bus. And we see this path here where students avoid the sidewalk and run straight across the lawn in order to catch the bus. And we end up with compacted soil, because when you've got to catch that bus, it doesn't matter if it's a rainy day or a dry day, you're going to run across that lawn. But even as farmers, when you're making that dash across your fields, you need to think about how we can protect and preserve that space so all of these biological functions that we rely on in soil can happen. If you're really curious about soil compaction, I wrote a little bit about soil compaction in this awesome new textbook that's freely available from the Canadian Soil Science Society. There's a whole chapter in there, chapter 15, on soil health and management practices that might be of interest to you if you're attending this session today. Okay, now with my professorial part done, where I've talked about some of the foundation elements and hopefully connected the whole program that we have for this morning, I'd like to come back to these 5 principles and tell you some stories about experiments we've been learning from at the University of Manitoba that look at some of these important principles for building soil health. We have tools to manage soil health, and the experiments that I'm going to be talking about really rely on some of these important tools that we have in our toolbox to utilize as managers to manage soil to improve soil health. So I want to talk about, firstly, crop rotation, right? That a reliable answer on Gary Martens's midterm in crop production. As a new professor in plant science, I arrived in Manitoba at a time when soybeans were increasing. I'm just going to jump forward to this slide. When soybeans were increasing. So I became a professor at the University of Manitoba in 2011. Right at this time, we were having this large increase in production of soybeans across Manitoba. It was an exciting time to be here. Things were changing. There were new challenges to look at. And I was very interested in the opportunities that soybean bring to our rotation, in terms of diversifying the predominantly wheat and canola rotations that we have. Adding legumes brings that important element of diversity. So crop rotation studies take a while, and I was lucky to have some early support from the pulse growers to establish a long-term crop rotation study where we looked at the frequency of soybeans in the rotation. And the work that I'm sharing with you now was primarily done by my graduate student, Shaturi Weerasekera. And so I want to acknowledge her in all the data that I'm going to share with you. This crop rotation study was started in 2014, and it occurred at 2 locations at the Ian Morrison Research Farm near Carmen, as well as at Richardson International's Kelburn Farm near St. Adolphe. So we had 2 very contrasting soil types. At Carmen, we had a clay loam, and then at Kelburn, we had a really heavy clay textured soil. And so what did I want to study? I wanted to look at how the frequency of soybean in the rotation influenced productivity of soybeans, but as well to be able to track and look at changes in soil health over time. So having stable long-term funding to do research is really hard, and it's really amazing but it was really amazing to be able to run this study for 8 years. And during those 8 years, we had 2 4-year rotation cycles where we looked at continuous soybeans. So we ended up growing soybeans for 8 years straight. Okay, we'll talk a little bit about how that was in a moment. And then we looked at soybeans in rotation with 2 other really common crops, canola and corn. This corn-soybean rotation helped me connect the research that we were doing in Manitoba with the broader literature on in the Midwest, where corns and soybeans are a major rotation. And then number 4, rotation 4, is really sort of our Manitoba rotation, how soybeans fit into the diversified suite of crops that we grow in Manitoba, including wheat, canola, corn, and soybean all together. These short soybean rotations are really of interest to me, and as exciting as it is to bring soybeans into our rotations, they also sort of spoke to the concern that I had at the time, which was if we're growing a lot of soybean is a low residue crop, how is that going to balance and influence soil health? So knowing that soybean is a low residue crop, I grew it with another what we consider low residue crop, canola, and then the highest residue crop that we have of corn. So most of the data I'm going to talk with you about this morning coming from this experiment is coming from this 8th year of the study in 2021. So after we had 8 years of these rotations at 2 locations in Manitoba. So my first objective was really to look at how rotation influences soybean performance, and we did that through a number of measures. Yield, of course, was important, as well as how plants grew, so looking at plant productivity and biomass. And we also looked at biological nitrogen fixation, so how, like Darryl was talking about, those nodules being that secret ingredient for this crop being climate friendly. Today, with the time that I have and the other studies that I want to get to, I'm really just going to focus on soybean productivity, but know that there's this landscape of other measurements that are supporting this study as well, and connect to some of the other work that collaborators who came to this long-term study and also were able to look at some other objectives related to pathology and then to the soil microbiome as well, and rhizobia, bacteria in the soil. So what did we learn after growing soybeans in all these different ways over an 8-year period? Well, we were really in a soybean honeymoon phase, right? I thought that our really heavy soybean rotations were going to collapse after a few years. And I think the results of this study are really consistent with what has made this crop popular in Manitoba, is that when we don't have a lot of pathogen pressure, we can grow soybeans reliably over many years and in many different spaces in our rotations. So even after growing soybeans continuously for 8 years, we never really saw the collapse of our soybean yield. At the Kelburn Research Farm site, we had absolutely no difference in our soybean test crop yield in 2021. And at Carmen, the only place where we really saw a yield start to dip was in our 8 years of continuous soybean. So I'm not going to say that these results are going to happen everywhere all the time. As a researcher, you're always thinking like, oh, my data is going to, provide this cautionary tale about how you need to balance crop rotation. But I think the data is honest in that it's consistent with, as long as you've got water to fill bean pods in August, soybeans have a good place in Manitoba rotations. So even though we only started to see a yield dip after 8 years, collaborators Fouad Daif and Dr. Almadimajan, who is now at Agriculture and Agri-Food Canada as a pathologist in Moradin, they were tracking soybean pathogens and identified 3 new soybean pathogens from my continuous 8 years of soybean rotation. So even though we didn't see the collapse of yield, there's some cautionary signs here that growing continuous soybeans is not really the way to go, unless you're a pathologist wanting to identify new pathogens. We also found some interesting results collaborating with Dr. Ivan Oresnik at the University of Manitoba in microbiology, who looked at the dynamics of Brady rhizobia populations in the soil. And that was an important question at this time because we didn't know how our hard winters were going to be influencing the ability of Brady rhizobia to overwinter. And so his work showed that the frequency of growing soybeans in rotation does influence the ability of Brady rhizobia to overwinter. As well, we see changes in those populations as we sample through different periods in the growing season. So there's some interesting things going on with these Brady rhizobia populations influenced by rotations. Getting to what I think is core for this session that we're talking about this morning on crop rotations, this experiment was a place where I wanted to take some new emerging soil health measures to our soils in Manitoba to see if we could detect differences in soil health with the different rotations that we were following. So in this study, we were able to look at 3 different categories of soil health measurements. The first was related to carbon, so total carbon, as well as active carbon, which is sort of a new concept. If this is a pie chart here of our total carbon pool, active carbon is just a sliver of the total carbon that we measure in soil, but it's the carbon that microbes can readily break down. It's the carbon that's going to be feeding microbes, and the total carbon in the pool has a large passive component that's really important for other functions of soil, like soil structure, soil water retention. So we wanted to see how crop rotation, and again, that input of crop residues, influenced active carbon. Another measurement for soil health that was sort of new, and we hadn't really had a lot of work done in Manitoba yet, was the concept of soil enzymes. So it's really hard to have these tools to measure microbes in soil, but one way that microbes sort of leave a message is when they die, their bodies kind of leak all their innards out into the soil. And their innards have a lot of soil enzymes, or enzymes in them that then get left in the soil. And so we can go and extract those enzymes as a fingerprint to see how active microbes have been in soil. The last thing that happened with these soil samples, again with Ivan Oresnik, was we looked at microbial populations of bacteria and fungi. And we would need an entire morning to really talk and unpack those. But know that that is a piece of what we were able to look at in this study. So on the carbon side, again, just like my soybean test crop, we didn't move much. I wasn't really expecting to move much in total carbon because of the timeline of this study. I mean, 8 years is a long time, but there really weren't big differences other than maybe our rotation where we included corn in the rotation in terms of the amount of carbon that we were inputting back into the soil. So we really only saw differences that we knew already at the beginning of the study between our 2 sites, and we had no significant differences in the total carbon pool. We also saw no differences in our active carbon pool, which was a bit of a surprise to me, but I guess that's what science is. It's full of surprises. We'll talk again about active carbon and some other experiments where I am seeing differences in active carbon. So it's a measurement that I'm still interested in, and we still need to be tracking our total carbon pool over time. How about some of these new and interesting soil enzymes? We looked at 3 different enzymes. The first one is really focused on carbon cycling only. The second one tells us about carbon and nitrogen cycling in the soil, and the third one focuses on phosphorus cycling. And we didn't really see any differences in this experiment with the third enzyme here that's focused on phosphorus cycling. So I'm going to focus on these two where we saw differences related to carbon and nitrogen cycling. So although active carbon and total carbon didn't really move much with our different crop rotations, the enzymes told us a little bit more about what was going on in that microbial community in response to the frequency of soybean in rotation and the other crops that were contributing carbon during the periods or the years we weren't growing carbon. So this first enzyme here, beta-glucosidase, looks at mostly carbon cycling. And we can see here that we had higher carbon cycling in this rotation at Carmen where we had corn in the rotation. So I think we're seeing a response to that higher input of corn biomass in our rotation. Then at the Kelburn site, we really didn't see a response to our higher carbon input rotations like the corn and soybean rotation or the rotation that had both wheat and corn in the rotation. But we did see a drag where we had less residue returned to the soil in our continuous soybean rotation. Looking at the second soil enzyme that tells us about both carbon and nitrogen cycling, there's an interesting split here that occurred at both sites. We can see sort of divide here between our really low residue rotations of continuous soybean or canola alternating with soybean to the two rotations that had more carbon input from wheat and corn in the rotation. So that was at least consistent between the two locations. So as a scientist, you're like, okay, great. Something's happening consistently in my study. So I think these soil enzymes have merit, but what we really need now is more information about how these enzymes perform across a greater diversity of soils and how they connect, or are they able to predict differences in crop performance or crop productivity. And because my experiment didn't really have any differences in crop productivity, unfortunately this data set doesn't really get that started. So Steve, we've got more work to do. All right. So I've talked to you a little bit about diversity and crop rotation as an important tool in soil health. And some of my measures for soil health detected those differences, and some of them didn't. I do want to connect the concepts. No, I want to start again. So although soybeans weren't showing great promise in this crop rotation experiment as the key driver pushing the advancements in these soil health measures, I do think soybeans still have an important role to play in our rotations related to some of the other aspects of these 5 soil health principles related to specifically to reducing a disturbance or reducing tillage and limiting soil cover. So I'm not going to go into these studies in detail. Maybe you've had the opportunity to hear me speak about them, but I want to connect them with the ability of pulses and soybeans to contribute to these other soil building practices. One of them is related to residue management after soybeans. So, in my crop rotation study, I was saying, well, we need to balance our rotation with high and low residue crops. But the opportunity we have for improving soil health when we do grow low residue crops is that for those that are using tillage in their rotation, this might be a place in your rotation where you can selectively reduce tillage. So I did an on-farm experiment for 3 years funded by the pulse and soybean growers where we looked at farmers' standard management practices with tillage and compared it to direct seeding soybeans into undisturbed stubble. And we saw only at one of the 5 locations that we ran these experiments an increase in soybean yield or an increase in yield following soybeans after we direct seeded into the soybeans. So I think that there's data in Manitoba to suggest that as long as we've got great residue management after soybean harvest, right, we're spreading that stock and we've got good planters that can penetrate the soil and create a good soil seed bed, we can rely on soybeans to be one of our early places to reduce tillage. We can also think about residue management ahead of soybeans. And this was a topic, again, that was of great interest to me when I was starting out at the University of Manitoba. As we were seeing, we were waiting for the genetics to bring us early season soybeans that were well adapted to all of Manitoba, we saw farmers doing a lot of tillage ahead of soybeans to make this long season crop fit into their rotation. So in on-farm experiments, again, I looked with my graduate student Patrick Walter at different strategies to manage corn residue ahead of soybeans in rotation. And we looked at practices like strip tillage or vertical tillage and compared them to conventional tillage. And again, in this study, we saw that there were a lot of tools where we could reduce tillage, even for a crop like corn, and still have good soybean yields. So we can find strategies even ahead of soybeans to reduce our use of tillage. And again, that piece of having the equipment, tools that we need to make that happen is part of reaching these soil health outcomes. So carrying on this theme of low residue, like soybeans, and in the case of other pulse crops, edible beans as low residue crops, we also have opportunities to think about how we can bring more residue and have more continuous cover, which is another concept in building soil health. Again, with a graduate student, Virginia Jansen, we were able to look at fall rye cover crops ahead of soybeans and edible beans in Manitoba and bring a tillage component into it. So we looked at this fall rye system, both direct seeding as well as strip tilling into to fall rye cover crop residue. We also looked at those two treatments across a range of termination dates. When you're managing cover crops, one of the really important questions you have is, when do I terminate this cover crop ahead of my soybean crop? And in Virginia's study, we found that soybeans were a really good crop to start managing fall rye cover crops with. We had a much wider window for termination of fall rye cover crops, and we saw that both direct seeding or strip tilling into terminated fall rye cover crops were good strategies for having consistent soybean yields. As long as we terminated the soybeans up to 1 day after planting, we had no difference in our soybean yields between our cover crop and no cover crop strategies with and without strip tillage. Where we terminated the cover crops later, we did see a decline in soybean yields where we were not using strip tillage. Edible beans was a slightly different story. There we saw edible beans really not do well with cover crops when they were terminated late. Part of that is driven by the lack of fit with herbicides that can terminate fall rye cover crops after planting edible beans. But if we're planting or terminating fall rye cover crops 2 weeks before planting, that is still a safe window, and edible beans being a later seeded crop, we probably have that window to be able to terminate fall rye cover crops earlier. So I think, again, soybeans and pulse crops in rotation, because they can fix their own nitrogen, is a really great entry point if you're looking for a place to bring overwintering cover crops that can help cover and protect soil, as well as provide more days where we have living roots growing in the soil as a strategy that you want to use to advance your soil health goals. So the last piece, and probably the hardest piece of these 5 soil health principles, is the last one, integrating livestock. If we do have many areas of Manitoba where we have great opportunities for grazing livestock directly on fields, and I think more and more we're seeing farmers see the value and looking for these relationships between different farm types that are either specialized into grazing or those that are really solely focused on livestock production. There are also indirect ways of integrating crops and livestock, and we had a really great opportunity to look at this indirect integration of livestock through the application of manure at a long-term study that Don Flayton started back in 2008 at the University of Manitoba Lawley Research Station as part of the National Centre for Livestock and the Environment. So Don laid out a fantastic experiment way back in 2008 where he compared perennial crop history to annual crop history, a really important part of diversity that we can take a look at and learn from in the aspects of soil health. He also contrasted really different fertility strategies. So a control treatment that had absolutely no fertilizer applied to it with a synthetic fertilizer-only strategy. So we were just using conventional fertilizers to provide fertility. And then contrasting that with manure from both a solid dairy manure or a liquid pig manure as their strategy for meeting the nitrogen requirements of the crop. As the lifespan of this experiment continued, Mario Tenuta has picked up the mantle for this experiment, and he's imposed some new treatments where we're looking at different soil management strategies. And another really important aspect, I think, of this study is the inherent soil type that's very unique. It's a heavy clay soil, so it has all the challenges, all the soil health challenges of being a heavy clay soil that's hard to manage. And yet, clays have this amazing capacity to build soil health really quickly because of the amazing properties of clay if we manage them well. So we had the opportunity to work with soil samples from this long-term study where we had these manure and fertility treatments applied from 2008 to 15. After 2015, there was no more soil fertility treatments applied, or sort of manure treatments applied. So we're really dealing with this legacy of this 8-year period where we applied manure. And then this really long-term history of having perennials versus annual crops only in the rotation. So it was a really fantastic place to pull on some long-term management to see if our soil health measurements could detect differences in management. We knew they were there in this experiment, some really extreme differences, some really long-term differences in soil health or in soil management over the time. So this research was conducted by my graduate student, Jacob Collodi, and Mario Tenuta was really essential in helping Jacob function as a graduate student during the pandemic when no one could be around. And Mario was there, and so I really want to acknowledge his role in this study. And what we wanted to do was take a soil health scorecard called the Cornell Soil Health Test and see whether it could detect differences in our soils in Manitoba. So this is a series of different types of measurements that both encompass both the physical, the biological, and the chemical properties of soil. This isn't a scorecard or a set of experiments that we could do on every single treatment in the study, so we went to this panacea of treatments that Don laid out in 2008 and selected 4 contrasting treatments. Again, our control treatment that had no amendments applied to it, our fertilizer only to meet the crop requirements, and then the solid dairy manure and the liquid pig manure. So we picked those treatments out where we had both perennial crop history and annual crop history. So what did this look like? The Cornell Soil Health test, as I mentioned, has different buckets of indicators that fall into different categories. So we looked at, again, some of these things that we were studying in Chituri's study with the crop rotation study, active carbon and total carbon. We compared them to some other standard methods that have been around for a long time, like potentially mineralizable nitrogen, soil respiration, as well as a new measure called soil protein. So again, when microbes die in the soil, they kind of leak their innards out into the soil, and so we're able to detect protein, or the protein that they exude and then build soil aggregates with. Then some also really traditional but time-intensive measurements of measuring soil compaction, water infiltration, and aggregate stability, as well as some really standard chemical properties that you can get on any standard soil test: organic matter, pH, electrical conductivity, and then a suite of nutrient measurements. So these are different buckets of measurements and these different properties. What we really wanted to know is how are we going to rearrange these measurements into indicators that were responsive to the management that we knew was there versus the management or versus indicators that weren't really able to pick up any response in our soils in Manitoba. So this is our big slide. This is our big takeaway. If you are interested in these, what measurements were responsive here. So we have this list here of indicators that could detect differences between our perennial versus annual crop history and between the different fertility strategies, manure versus no inputs versus a fertilizer-only strategy. So we had this group of unresponsive of indicators that might still be important for some soils, but weren't really discriminating for the properties that we were looking, or the treatments that we were looking at. So let's unpack some of these indicators, and maybe I will do this quickly because I think I'm running out of time. So I promised you that active carbon was going to matter, and here is my slide showing that active carbon matters, and it is discriminating between these different strategies for fertility management. So again, coming to the role of integrating livestock, even if it's just about bringing manure to your grain production fields, we can see that solid dairy manure, as a manure that has a lot of organic matter into it because of the bedding that gets incorporated with it, really stood out as having higher active carbon, even as a legacy, right? The last manure treatment that occurred in this experiment was 2015, and Jacob went out and collected these samples in the fall of 2020. So even 5 years later, we could see this active carbon bump from the application of solid dairy manure. Not statistically different from our synthetic or controlled treatments, but numerically higher was liquid pig manure. And we see these same results, similar results play out in our total carbon as we would expect. Looking at some of these other measurements related to nitrogen cycling, and also still kind of tied to carbon, in that we know that the manure treatments that we're seeing responses to are also contributing carbon to these systems that microbes are utilizing as in ratio with nitrogen, is acyl protein. So again, the proteins that are spilled from the innards of microbes. So it's a sort of an indicator of microbial activity. And then potentially mineralizable nitrogen, where we take these soils and we incubate them and look at what nitrogen can be mineralized from these soils that you could expect, over the next growing season. So again, we see solid dairy manure really stand out, and I think that is because it is both contributing nitrogen and a carbon source to these long-term treatments. So taking a look at some other measurements that are connected to microbial function in the soil is soil respiration and potentially mineralizable nitrogen. But looking at a different slice of the experiment, and that is our perennial versus annual crop history treatment. So you'll see that my boxes here slim down to just the 2 treatments that we had. So if you were writing Gary Martin's crop production exam, after you put in crop rotation, the second answer that would get you some marks was perennials in rotation, right? Those perennial crops have value. We know they have value. They're another way that we can integrate at least livestock, at least as a source of producing livestock feed. Even if the livestock aren't on your farm, those perennials have value, and we see microbes respond to it in that they leave these roots behind, and those microbes get to use them as a source of energy and nutrients to thrive after the perennials are gone. And we also see that they provided an increase in potentially mineralizable nitrogen in these experiments. One of the measures that was new for Jacob and I to figure out was looking at aggregate stability. And I think aggregate stability is one of these soil health measures that really help integrate a lot of variables together. It is classified as a soil physical property, but it really relates to the biological functioning of the soil and how the biological microbes in the soil help us to build those soil aggregates, to create those homes, and have that space for air and water in soil. And okay, so we see here, no surprise, that the soils that had perennial crop history did create more stable aggregates that can withstand more erosive forces. So what you see here in this picture is a Cornell soil health infiltrometer that we suspended on a tripod above a funnel that has aggregates that we collected and dried from the experiment. So this infiltrometer here creates a series of water drops analogous to rain falling on the soil surface. So it was a really good test to look at indicators of soil health that connect maybe to protecting the soil from soil erosion and holding pores open at the soil surface so that water can infiltrate into the soil. So again, in summary for this study, from all the hard work that Don Flayton did to establish this study and all the hard work that Mario's doing to continue this study, we really see the value in long-term studies in being able to go and test drive these new measurements of soil health, because we did find that even though this study or the Cornell Soil Health Test was developed and calibrated from soils from a very different region from Manitoba, where we have these really young, high organic matter soils, they were able to detect differences in our historic fertility treatments as well as our cropping treatments. Thankfully, we have other scientists like Steve and also a scientist or professor, Kate Congreves at the University of Saskatchewan, who has been doing more work to look at how we can calibrate these useful soil health tests to our soils in Western Canada. And Kate recently published a paper with her graduate student, Athena Wu, in 2001, looking at what she's calling the Saskatchewan Assessment for Soil Health, so taking and modifying the Cornell Soil Health Test to soils in the prairies. And this is an open access journal article in the Canadian Journal of Soil Science. If you're interested in learning more about responsive soil health indicators in the prairies, this is a really good read. So with that, there is a lot that we've been able to learn at the University of Manitoba from our long-term studies about soil health and how the crops that we grow in rotation can help us contribute and build to soil health. This work does not happen alone. I wanted to thank my graduate students again and the technical support of my lab team, as well as the funding sources that helped make all of this work that I present to you possible today. So with that, I will turn things back over to Daryl and to the rest of the series that we have this morning.
There'll be questions now.
Okay.
Okay, thanks, Yvonne. So Yvonne, I think what you did was give us a real quick run-through of the complexity of soil health, of a concept like soil health, and just how many parts are moving. Subsequent talks will get into some of those specific parts. But I want to know if anyone in the audience has a question for Yvonne based on the sort of bird's eye view she's given us. Yes, sir.
So the question, as I understand it, is about the row spacing of the soybeans and how that influenced the soil health results or the soil health tests that we did from the soil. So in the experiment, we were growing soybeans on 30-inch row spacing versus solid seeding. I take that back. No, we did grow in this experiment. We did grow them on 15-inch row spacing. Okay, so I don't think that it would influence a lot of the soil health indicators in terms of soil enzymes or the total carbon of the soil, because we were using tillage in that experiment to incorporate residues. I think that wide row spacing versus narrow row spacing would influence other soil physical indicators like infiltration, because you have that inter-row area that's often fallow for a long period until we have row canopy closure, where you can have a lot of closing of those pores. I'm doing some intercropping work, looking at how to intercrop cover crops in that inter-row area for wide row crops. So I think that there are tools that we could look at in the future on how to manage that inter-row area for wide row crops. And I think that When you're using tillage in the system, I wouldn't expect many differences between narrow versus wide rows, but in a conventional till system, in a zero till or minimum till system where you are reducing your soil disturbance and keeping those soil pores and columns and macropores intact, you could see differences emerge over time. But I haven't studied or measured that yet.
Okay, any other? Yes, ma'am.
Great question. So the question is about the timeline to see soil health differences for growing perennial crops in rotation. And I have not studied that. I don't know. I don't know, Steve, if you've looked at that. I can tell you from the long-term nickel study that the perennials in rotation in that experiment, I'm trying to remember the exact dates, were sort of grown in 2 phases. And I think that they were 3 to 4 years in length, but there was reestablishment of those stands. And so in that experiment, we did have a long period of perennials grown, but they were grown in sort of 3 to 4 year stands. Great question though, and it'll be great to have some data to look at that.
Okay, the back and then here. Yes, sir. Get that?
Okay, so the question was about how microbes in the soil connect with the plants that we grow, and have we been studying the soil microbiome? I have not been studying the soil microbiome, but I have been collaborating with a researcher, Ivan Resnick, in the soybean crop rotation study, who was looking at the diversity of soil bacteria in that study. And we have a soil microbiologist here, Matthew Bacher, who is going to be on the panel this afternoon. And I think I'm going to defer to Matthew about the state of what we know on the soil microbiome and connection to plants.
Sure, so stick around and you'll hear about that. And there was a question here. Which perennials did you use?
Okay, which perennials were used in the nickel long-term experiment? It was alfalfa in the rotation. Yep.
Okay. Yes, sir.
So, what soil health test do you like? What test gives your best measure of soil health?
Thank you for that question. What is the best measure that I should use now, today, to measure soil health? Do we have our panel this afternoon? Could we talk this out there? I think that this is a hard question with where we're at right now. And so I recognize that many of you sitting here in the audience want me to say, like, all you have to do is go and measure this one thing, and you're going to know whether you have good soil health or not, or if what you're doing is changing the soil health of your soil. And just like, you know, the 1960s, the 1950s were periods where we had a lot of development and methods for soil nutrient testing. We are in that exact time right now for soil health. And so I would say, if you are a person who is wanting to measure and track your soil health test right now, the things that you have available for you to go ship off that soil health test and get that soil health test right now is limited, and there's some frustration there. There are some really good things that you can turn to and rely on, like total carbon. That's a good measure. That's a long-term measure. We can now, you can now send samples away for active carbon. I think AgVise has that. It's called POX-C on their list of recommendations. I think there's also just a lot of visual assessment that you can do related to soil physical properties, like infiltration, like aggregate stability, taking a soil aggregate and putting it in water and seeing how stable it is over time. Are those fantastic tools that you have in your toolbox right now? No. But I am very optimistic that in the next 5 to 10 years, what you are going to have available to you is going to change. And as I mentioned, scientists like Kate Congreves at the U of S, who are further along in looking at a comprehensive soil health assessment, if we can keep supporting that work through funding for research, I think that we're going to have more tools and locally soil health tests that are calibrated to our soils with time.
Thanks, Yvonne. I'll just answer that a bit too. One of the reasons we're investing as Manitoba pulse and soybean growers in this is that we think in a few years we'll be able ourselves to perhaps help farmers test their soil for soil health, just sort of as a service. Starting there, if it becomes a commercial thing, great, but sometimes these things just need to be pieced together and trialed with groups like commodity groups, with the farmer's interest as the first point of concern. And I think that's why we're funding some of this stuff. Just to end off, I have one little question here, Yvonne. So at the beginning of your presentation, you had a photo of a close-up of a row of crop, and you said that that's a healthy looking soil. But as you've told us, and as we'll find out here even more so in subsequent talks, a lot of stuff about soil health you can't see because it's down there. But with all the different practices we're doing, no-till, crop rotation, and all these things, is there one or two things that kind of stick out that farmers might be changing or doing to their soil and they don't even know it because they can't see it? Is there something there that's really kind of something that we're gonna one day wake up and say, like micropores used to be that. You used to till your soil a lot without really recognizing the damage it was doing to the porosity of the soil. And now we know that. Is there something like that that's going to come up, or is that just something we need to keep researching and digging down and finding out?
Okay, this is the exam question right here, Daryl.
Who's the professor?
I'm like freezing up on a good answer.
Okay.
But what I would say is I think farmers know which soils are healthy and which soils are not. I think you know those areas of the field where you like drive over them, or you see your crops growing in them, or you go out like Frank Prince did in that video that we watched at the very beginning, you put that shovel in and you can smell that soil is doing well. You also know the areas that you're worried about. And so I would say that I think that rely on your instincts and rely on your senses to follow soils. In the absence of these amazing tests that we want to have right now, I think what the big challenge is right now is knowing, okay, I want to do something about this area. How do I know if what I'm investing, like if I'm going to change, if I'm going to invest in this change, because it's not easy, because if it was easy, we'd all be doing it already. If it was easy, if it's going to take work, how do I know that that's paying off for me? And I think that's one of the things we want to get into in our panel this afternoon.
So final thing, what's that smell called? Their aromatic smell.
Do you know the name, Matthew?
Geosmin.
It's related to the fungi in the soil.
Yeah.
When you have that good earthy smell.
Did you know that? It's geosmin. Next time you smell that when you're cultivating and things like that, I think we should have a class where we take a bunch of people from the city. We say, we're going to teach you about agriculture, and we just bring them to a field somewhere around Brandon, and we stand them there, and then they're wondering what's happening next. We say, breathe in. And then we can tell them all about geosmin and healthy soil. Yvonne, thank you very much. For all our speakers today, we're making a contribution to the farmer mental wellness program on their behalf. So thanks, Yvonne.
Yeah, thank you, Darryl.